Hi friends, antimicrobial world welcomes you. In this video, we are going to see what are endocytosis, phagocytosis and pinocytosis. Endocytosis. Endocytosis is the cellular process by which cells absorb external material by engulfing it with the cell membrane. That is, extracellular molecules are taken up into the cell to form membrane-bound vesicles called endosomes. In simple words, we can say endocytosis is the process by which substances are engulfed into the cell. Endocytosis is a general term and it is usually subdivided into phagocytosis and pinocytosis. Next, we are going to see what are phagocytes and phagocytosis. Phagocytes are also known as phagocytic cells. Phagocytes are cells that have the ability to ingest and sometimes digest foreign particles such as bacteria, dust, dead cells, etc. Natural defense against the invasion of blood and tissues by microorganisms and other foreign particles is mediated to a large extent by phagocytic cells, which ingest and destroy them. Phagocytic cells were originally discovered by Mechnikov. Mechnikov classified phagocytes into microphages and macrophages. Microphages are polymorphonuclear leukocytes. Macrophages consist of the histiocytes, which are the wandering amoeboid cells seen in tissues, the fixed reticuloendothelial cells, and the monocytes of the blood. 1 liter of human blood contains about 6 billion phagocytes. Phagocytes of humans and other animals are divided into professional and non-professional groups based on the efficiency with which they participate in phagocytosis. The professional phagocytes are a specialized group of cells which can accomplish phagocytosis with high efficiency. The professional phagocytes are monocytes, macrophages, neutrophils, tissue dendritic cells, and mast cells. The professional phagocytes have molecules called receptors on their surfaces that can detect harmful objects such as bacteria that are not normally found in the body. Professional phagocytes are responsible of removing microorganisms and of presenting antigens to lymphocytes in order to activate an adaptive immune response. Next, we'll see what are non-professional phagocytes. The non-professional phagocytes can accomplish phagocytosis with low efficiency. Non-professional phagocytes do not have efficient phagocytic receptors and cannot ingest microorganisms but are important in eliminating apoptic bodies. The non-professional phagocytes are fibroblasts, epithelial cells and endothelial cells. Next, we are going to see how phagocytes reach the site of infection. During an infection, chemical signals attract phagocytes to places where the pathogen has invaded the body. These chemicals may come from bacteria or from other phagocytes already present. The phagocytes move by a method called chemotaxis. When phagocytes come in contact with bacteria, the receptors on the phagocyte's surface will bind to them. This binding will lead to the engulfing of the bacteria by the phagocyte. Some phagocytes kill the ingested pathogen with oxidants and nitric oxide. After phagocytosis, macrophages and dendritic cells can also participate in antigen presentation. This process is important in building immunity. Phagocytosis. Phagocytosis is a cellular process by which phagocytes ingest or engulf extracellular particulate materials such as bacteria, invasive fungi, parasites, dead host cells, cellular or foreign debris to protect the body. In phagocytosis, a cell's plasma membrane expands around the particulate material, which may include all pathogenic microorganisms. Most phagocytosis is conducted by specialized cells such as blood monocytes, neutrophils, and tissue macrophages. The mechanism of phagocytosis. Bacteria are progressively engulfed by the cell membrane into a vacuole called the phagosome. Phagosome fuses with the lysosome found in the cell to form the phagolysosome. The bacteria are subjected to the action of the lytic enzymes in the phagolysosome and are destroyed. We must note one important point here, that is, some bacteria resist intracellular digestion and may actively multiply inside the phagocytic cells. Phagocytosis in such instances may actually help to disseminate infection to different parts of the body. Salmonella are more resistant to toxic form of oxygen and to defensins, toxic peptides that kill bacteria. Some bacteria such as Shigella flexneri and the spotted fever rickettsia escape from the phagosome into the cytoplasm prior to the phagosome fusing with the lysosome. Neisseria gonorrhoeae produces proprotein that prevents phagosomes from fusing with lysosomes, enabling the bacteria to survive inside phagocytes. We know that neutrophils use toxic oxidants to kill bacteria. The carotenoid pigments give Staphylococcus aureus its golden color and group B streptococci its orange tint. The carotenoid pigments shield the bacteria from the toxic oxidants. Some bacteria are able to kill phagocytes. Bacteria such as Staphylococcus aureus and Streptococcus pyogenes produce the exotoxin leukocidin, which damages the cytoplasmic membrane of the phagocyte. Bacteria such as Shigella and Salmonella induce macrophage apoptosis, a programmed cell death. Next, we'll see the importance of phagocytosis. The importance of phagocytosis in protection against infection is evidenced by the enhanced susceptibility to infection seen either when the phagocytic cells are depleted, as in agranulocytosis, or when they are functionally deficient, as in chronic granulomatous disease. Up to this, we learned what is endocytosis and phagocytosis. Next, we are going to see what is pinocytosis. Pinocytosis, the process by which cells take up fluid from the surrounding medium along with any molecules contained in it. Next, we are going to see the difference between. At first, we are going to see the difference between endocytosis and exocytosis. Endocytosis is the process by which substances are engulfed into the cell. Exocytosis is the process by which substances are released from the cell. In endocytosis, extracellular macromolecules are taken up into the cell to form membrane-bound vesicles called endosomes. In exocytosis, macromolecules are moved from the interior to the exterior of the cell. Next, we are going to see the difference between phagocytosis and pinocytosis. Phagocytosis is a cellular process by which phagocytes ingest or engulf large solid particles such as bacteria, dust, dead cells, etc. into the cell for immune purposes. Pinocytosis is the cellular process by which cells take up fluid from the surrounding medium. Finally, we are going to see the difference between professional and non-professional phagocytes. The professional phagocytes are a specialized group of cells which can accomplish phagocytosis with high efficiency. The non-professional phagocytic cells are group of cells which can accomplish phagocytosis with low efficiency. The professional phagocytes have molecules called receptors on their surfaces that can detect harmful objects such as bacteria that are not normally found in the body. Non-professional phagocytes do not have efficient phagocytic receptors. The professional phagocytic cells are in charge of eliminating microorganisms and of presenting them to cells of the adaptive immune system. The non-professional phagocytic cells are important in eliminating apoptic bodies. The professional phagocytes are neutrophils, monocytes, macrophages, mast cells, and dendritic cells. The non-professional phagocytes are fibroblasts, epithelial cells, and endothelial cells. I am concluding this video here and see you soon in my next video. Thank you, friends.